Hello students, how you all are? I hope you all are doing very good. So let's start the chapter today, and the chapter for today is Transport in Plants. That is the first chapter of plant physiology. And grab your NCRTs and read it along with me and learn the chapter along with me. So let's start the chapter. And the chapter starts with the types of transport. In the plant, two types of transport are there: short distance transport and long distance transport. Now, what is the meaning of short distance transport? The short distance transport can be intracellular, that means within the cell. From one part of the cell to the other part of the cell is intracellular short distance transport. Then it can be intercellular, that means between two cells. And this kind of transport occurs with the help of a bridge-like structure between the two cells, and that is plasmodesmata. Now, across the membrane, this is also short distance transport. If some molecule has to enter into the cell, that is entry into the cell across the membrane, and exit from the cell, that is also across the membrane. That means short distance transport can be intracellular, it can be intercellular, and it can be across the membrane, that is entry into the cell or exit from the cell. Long distance transport, on the other hand, is also called as translocation, which occurs through xylem and phloem. So these are two kinds of transport. Let's first discuss the short distance transport. What are the forces in the plant that are responsible for the short distance transport? Four major forces: simple diffusion, facilitated diffusion, active transport, and cytoplasmic streaming. These two forces are the passive forces, no expenditure of energy, and these two forces are active forces, that means expenditure of energy. These four forces together are responsible for short distance transport in plants, while for long distance transport, the forces are mass flow, root pressure, and transpirational pull. Clear? Let's move on to further things. Now there is NCRT line which says that if the translocation occurs via xylem, it is unidirectional, and if occurs through phloem, it is multidirectional. What is the meaning of that? In case of xylem, the source is always fixed. That means from where the water and minerals go, that is fixed, and that is soil. That means in the xylem, the water and the minerals will always go from the soil to the different parts of the plant, which are called as sink. So the source to sink relationship in case of xylem is always fixed. That is always from root to the different parts of the plant. That is why the translocation in xylem is always unidirectional. But in case of phloem, the source to sink relationship is not fixed. It changes season to season. Why? When the plant has full-fledged leaves, then in that case, the leaf will act as the source. That means the food will go from the leaf to the different parts of the plant or towards the root. But what will happen when the autumn season will come and the leaf will fall? The plants will have no leaf. So then the food gets stored in the storage organs like root. So in the autumn season, when the plant have no leaf, the food gets stored in the storage parts like root. And when the spring will come and the new leaves has to come on the flower, this food will be supplied to those parts, from root to those parts, right? So the source has changed now. So the source to sink relationship is not fixed in case of phloem. It changes from season to season. That is why multi-directional. Clear? Now let's discuss what are the materials that gets translocated through xylem and phloem. Water. Water will always be translocated through xylem, and that too only in unidirectional manner. Always in unidirectional manner. because water will always go from soil towards the different parts of the plant. Minerals translocated both through xylem and phloem. How? In xylem, the translocation of minerals is always unidirectional, always from soil to the different parts. But in case of phloem, the translocation of minerals is multidirectional. Now you will say that, ma'am, we have read that the translocation of minerals occurs through xylem, and you're saying that the minerals are translocated both through xylem and phloem. Are you saying it right? Yes, I'm saying it right. Minerals are translocated both through xylem and phloem. But in case of xylem, the source will always be the root. But in case of phloem, what's the case? Let's understand. If I say there is some leaf on the plant which is old, and now it has to detach from the plant, it has to get separated from the plant. But before it gets separated from the plant, it donates all its material to the younger parts, to the younger leaves. That means before going away or before the death, it gives all its property to the younger parts, to the younger children in its will, right? So when this old leaf, when this old aging leaf or senescent leaf will separate from the plant, it will give its mineral to the younger leaf, and then the translocation of minerals does not occur through xylem but through phloem. So the minerals from old part to the younger parts, they get translocated via phloem. This is how minerals gets translocated both through xylem and phloem. Clear? Now food always through phloem, and that too in multi-directional, or you can say bidirectional manner. Somewhere in NCERT it is written bidirectional or multi-directional, because source to sink relationship is not fixed. Then comes the hormones. The different kinds of hormones in the plants are also translocated through xylem and phloem, and it is written in NCERT that it is done sometimes in strictly polarized manner. Sometimes it happens in strictly polarized manner. That means strictly in particular direction, right? So these are the things which gets translocated through xylem and phloem. Clear? Okay. Now let's get back to the four forces which are responsible for the short distance transport. And the first one was simple diffusion. Simple diffusion is a kind of movement of molecules which is not restricted to only the living systems. You can see diffusion everywhere, right? So it is seen both in living and non-living systems. Simple diffusion is a passive process because it does not require expenditure of energy. Simple diffusion happens from high concentration to the areas of low concentration. That means this kind of movement is called as down the gradient movement or downhill movement or along the gradient movement. The kind of movement which happens from high concentration to low concentration, from the region of high concentration to the region of lower concentration, this kind of movement is called as downhill movement or down the gradient movement or along the gradient movement. Right? And for diffusion to happen, like for electricity to flow, there must be potential difference between the 2 points. So for diffusion to happen, there must be difference between the concentrations between the 2 points. That means there must be concentration gradient between the 2 points. Then only diffusion will occur. Clear? Okay. Now, during diffusion, the movement of molecules is random, that is not organized. They move in an unorganized manner, and the movement of one molecule is different, is independent from the movement of the other molecule because they move randomly. And the more the disorderliness in the system, the more the entropy in the system, the more would be the rate of diffusion, because during diffusion the movement of molecules is random, and entropy supports randomness. Okay, so diffusion is an entropy driven process. Then diffusion is very common in gases, right? It's then in liquid. But have you ever seen that there are 4 or 5 chairs in one corner of the room, and there is a lesser concentration of chairs in the second corner of the room, and when you will come in the morning, you'll come back in the morning in the room, you'll see that because the concentration of chair was high in that corner, so the chair will move by itself towards the corner where the concentration of the chair was less. Will it happen? No, because the diffusion of solids is not likely, but the diffusion can happen in solid. This room is a solid, and diffusion of gases can happen in solid, but of solid, no. So this is the very important line of NCERT, that diffusion in solid is more likely to happen, can happen, but diffusion of solids, no. Okay, so diffusion is very common in case of gases. Now, what are the factors on which the rate of diffusion depends? Obviously, the very first one is the concentration gradient. The more the concentration gradient, the more would be the rate of diffusion. Then permeability of the membrane. If I talk about a living cell and something has to diffuse into the living cell, then obviously if the permeability of the membrane towards that molecule would be high, the rate of diffusion would be high. If the membrane will not allow the movement of molecule, then how diffusion will happen? So yes, permeability of the membrane matters. Temperature. The more the temperature, the more would be the kinetic energy of the molecules, and the more would be the rate of diffusion. Pressure. If you apply external pressure to the system, then you will tend to increase the energy of the molecules and you will increase the rate of diffusion. But if the density of the molecules which have to move is high, that means if they are fat, then they will be, they will move with lesser speed. The rate of diffusion would be less. And if they are less denser or they have less molecular weight, they will move with great speed. So these are the factors which are important for deciding the rate of diffusion. Now let's come to another kind of diffusion, that is facilitated diffusion. Now to understand this phenomenon, let's go to the nature of plasma membrane first. The nature of the plasma membrane, you must have heard that plasma membrane has bilipid layer, right? So that means what kind of nature plasma membrane has? The nature of plasma membrane is hydrophobic. That means something which is soluble in water or water itself. Plasma membrane does not like those kind of molecules which are water soluble or water itself. So what is the nature of plasma membrane? Lyophilic, lipid soluble, lipid loving, and that is non-polar in nature. So all those molecules which has hydrophobic, lyophilic or non-polar nature, they can easily cross the plasma membrane without any problem, because they have the similar nature as that of the plasma membrane. But if I talk about the kind of molecules which have opposite nature to that of the plasma membrane, like the molecules which have hydrophilic nature, that means they are water loving and they are lipid repelling, they are polar in nature, or if it's water itself, then all these kind of molecules have opposite nature to that of plasma membrane. So plasma membrane will not easily allow the movement into the cell. So what will happen to these kind of molecules? Will they never be able to enter into the cell? So in this case, in the plasma membrane, there are certain proteins which are present, and those proteins are called as channel proteins or carrier proteins. These kind of proteins, they come for the rescue for these kind of molecules. What they say, listen, we are watching that you are struggling to come into the cell. It is saying to these kind of molecules that yes, we are saying that you are struggling, so we can help you out. The membrane proteins, the channel proteins, the carrier proteins are saying to these kind of molecules that we can help you out, and we are social workers, we are member of some NGO, we will not charge anything from you, but there is one thing, we have to say that we can help you to get transported into the cell, but there is one condition, that the concentration should be high, because we can do this work only from high concentration to low concentration. Only that is their condition. Otherwise, they do not charge anything from these molecules to get them transported into the cell. That means here diffusion is happening. Diffusion is happening because the transport is occurring from high concentration to low concentration. That is downhill, that is down the gradient, along the gradient. But here the diffusion is helped by or facilitated by carrier proteins or channel proteins or membrane proteins. They are helping these kind of molecules which have opposite nature to that of plasma membrane to cross the membrane without charging anything, without the expenditure of energy. But their condition is that they can do they can do this work only from area of high concentration to the area of low concentration. So the movement of these kind of molecule is helped by channel proteins or the carrier proteins which are present in the plasma membrane, right? So you can say that facilitated diffusion is also passive in nature. It often comes, the question often comes that because in facilitated diffusion membrane proteins are involved, so it is active in nature. No, even if the membrane proteins are involved, the process is passive because it is occurring from high concentration to low concentration, that is down the gradient. But yes, it is helped by some proteins present in the membrane, but they do not charge anything for doing that work for the molecules. Clear? So a special kind of diffusion where the membrane proteins, the channel proteins, the carrier proteins are helping those molecules which have opposite nature to the plasma membrane to cross the membrane. This kind of diffusion is called as facilitated diffusion, right? Okay, now let's talk about the channel proteins or the carrier proteins. They are social workers, right? And they are the integral part of the membrane. They are present inside the membrane. They do not require any energy for doing the work. So that is why the facilitated diffusion is passive in nature. And they are not associated with any ATPase activity. These channel proteins are not associated with any ATPase activity because they do not require ATP. And they do not have the capacity to set up the gradient. That means they can only do the work from high concentration to low concentration. They do not have the capacity to do it from low to high. They do not have the capacity to set up the gradient. They use only already set up gradient, that is from high to low concentration. Now these are the words of NCERT which are very important. What are the properties of the channel proteins or the carrier proteins which help in facilitated diffusion? They are very specific. That means any molecule will stand out of the cell and say that I have to enter into the cell. It will say no, you are not required into the cell. So they are very specific for the molecule it has to allow to go into the cell. Then they tend to saturate. They have their own limit. They cannot transfer thousands of molecules at one time. They have their own limit. After that, they saturate. They are very sensitive to inhibitors. If any chemical inhibitor is present in the plant, they are very sensitive to it and will stop working. And they are under hormonal regulation. The hormones regulate their activities. So these are the properties of the channel proteins or the carrier proteins. Now, porins. Sometimes in the plasma membrane, the size of the channel proteins is bigger, and those large sized channel proteins are called as porins. And where porins are found? Porins are found in the outer membrane of mitochondria, not the inner membrane. Outer membrane of mitochondria, outer membrane of chloroplast, and outer membrane of bacteria, most commonly the gram-negative bacteria, right? In the plasma membrane, there are water channels also, which allow the movement of water into the cell. And for the formation of these water channels, there are special membrane proteins called as aquaporins. Aquaporins. And the water channel in the plasma membrane is formed by 8 different types of aquaporins. Okay. Now let's move further. Now how these channel proteins or membrane proteins, they function when they transport the molecules across the membrane? By rotating by 180 degree. This is the membrane protein present in the membrane. This is the molecule outside the cell which is ready to be transported. So this molecule gets binded with the membrane protein. The membrane protein rotated by 180 degree, released the molecule into the cell and again come back to its conformation, right? It will get attached to the molecule, rotate by 180 degree, release the molecule into the cell and will again rotate by 180 degree. So this is how these membrane proteins function. Now, what kind of transport can be seen while the facilitated diffusion? It can be uniport, antiport and symport. Now what is the meaning of it? Uniport means when the transfer of molecule across the membrane is totally independent from any other molecule. Here the molecule A is getting transported across the membrane without any dependency on any other molecule, right? But here in case of antiport and in case of symport, the movement of molecule is dependent on the other molecule, right? So in first case, in uniport, the movement of molecule is independent of other molecule, but in case of antiport and symport, the movement of molecule depends on other molecule. What does that mean? When this molecule A has to go into the cell, then simultaneously the molecule B will come out of the cell. That means from this point the protein will be attached to A, from this point the protein will be attached to B, then it will rotate, it will take A into the cell and B out of the cell. So here the movement of two molecules is getting transported simultaneously, right? So transport of A is dependent on transport of B. A will go into the cell only when B will come out of the cell because they are opposite to each other. A does not want B into the cell, right? So here when A is getting into the cell, simultaneously B is getting out of the cell. This is antiport. But in this case, A is saying that, oh, I am afraid of going into the cell because maybe what I come across into the cell, I don't know. So I will take my brother, I will take my bouncer along with me. So A will always go along with B into the cell. That means they will be transported together, and this type of transport is called as symport. Okay, so these are three different kinds of transport. Clear? Now we have discussed two forces for short distance transport: simple diffusion, facilitated diffusion. Now the active transport. Obviously active transport, that means energy will be required. Why? Because in this kind of transport, this is wrong, sorry beta, the membrane proteins are involved and they are called as pumps. Okay, if this is a case that this is the cell and the molecule A has to be transported into the cell, but the concentration of A is low outside the cell and high inside the cell. That means A has to be transported from low concentration to high concentration. Then how it will happen? It will happen with the help of certain membrane proteins, and they are called as pumps. So if the material has to be transported from high to low concentration, this is done with the help of special membrane proteins called as pumps. And these pumps have the capacity to set up the gradient. They have the capacity to do this work against the gradient. That means from low to high concentration. And that is why they are associated with ATPase activity, because they need ATP for their function to work. And the pumps has the capacity to transfer the molecule against the concentration gradient. what is the meaning of against the concentration gradient? That means from low to high concentration, and this kind of movement is called as uphill movement. In facilitated diffusion and in diffusion there was downhill movement, but in active transport there is uphill movement or against the gradient movement, which is helped by pumps, right? The properties of pumps are similar to the properties of channel proteins involved in facilitated diffusion. They also have a limit, they also tend to saturate, they are also very specific, they are also sensitive to inhibitors, and they are also under hormonal regulation. Now, this is a table from NCERT, you can see here, this is very important. They have compared the three processes: simple diffusion, facilitated diffusion, and the active transport. It is saying that requires special membrane proteins. Obviously, simple diffusion does not require any membrane proteins, but the facilitated diffusion, channel proteins, and the active transport, the pumps. Selective, wherever the membrane proteins are involved, that process will become selective. So in diffusion process, no, it is not selective. This facilitated transport, yes. Active transport, yes. Is there any limit to the transport? Because in diffusion no membrane proteins are involved, so there is no limit in simple diffusion, but yes, in facilitated diffusion and active transport there is limit. Uphill transport means against the gradient movement, and against the gradient movement is seen only in case of active transport. Facilitated and simple diffusion, both downhill movement or down the gradient movement. ATP is required only in case of the active transport, not in simple diffusion and not in facilitated diffusion. Clear? Now, the last force for the short distance transport is cytoplasmic streaming or cyclosis. What is the meaning of cyclosis? It is the clockwise or anti-clockwise movement of cytoplasm in the cell. And this kind of movement is helped by special thread-like structures present in the cytoplasm called as cytoskeleton. And this movement of cytoplasm help in the intracellular movement, only intracellular movement, right? And this is active in nature. This requires ATP. Clear, beta? So we have discussed the short distance transport, and the forces were simple diffusion, facilitated diffusion, active transport, and cytoplasmic streaming, in which simple diffusion, facilitated diffusion, passive in nature, and active transport and cytoplasmic streaming, active in nature. Clear? So I hope up till here it's understandable to you. Let's go further now. Let's discuss the plant water relations now. Water is very important for plant, right? Because with the help of water, photosynthesis occur. Water acts as a medium for the various enzymatic reactions. Water maintains the shape and turgidity of the cell. So there is very, intimate relation between the water and the plant. Without water, plant cannot do anything. Approximately 75% of the water of the plant contains water. And in some plants, it can account up to 92%, like in case of watermelon. In case of herbaceous plants, the content of water is very high. So let's focus on water now, right? So when I am focusing on water, I have to explain a term that is called as water potential. Now, what is water potential? When the water is in its pure state, right, then the molecules of water have a certain free energy and a kinetic energy with which the water molecules can move from its higher concentration to its lower concentration. And the kinetic energy, the free energy of the water molecules with which they can move, is called as the potential of the water, and that is called as water potential. It is denoted by the symbol psi W, and it is measured in the units of pressure, that is Pascal or bar. So how can it define water potential? The kinetic energy of water molecule in their gaseous or liquid state with which water molecules move is water potential. Clear? If the water molecule is completely in its pure state, then we take a reference value, we take a standard value to explain the condition of water potential, and the water potential of pure water is 0, and that is taken as the maximum value because it is a reference value. And when we will make this water as a solution, then the water potential will decrease, it will become a negative value. So for pure water, the water potential is taken as to be 0, but under what conditions? Under conditions of standard temperature and standard pressure. You know what is standard temperature? 25 degrees centigrade. And what is standard pressure? The atmospheric pressure. So remember this point. Under standard conditions of temperature and pressure, the water potential of pure water is taken as to be 0. But what happens when I will add sugar to it, or when will I add salt to it? So sugar and salt are the solutes. So when I will add sugar and salt to this pure water, it will not remain as water, it will become a solution, and the water will be solvent. And when you have mixed the molecules of sugar and salt with it, you are decreasing the kinetic energy of water molecules. So the water potential of the whole solution will decrease. Say the water potential of pure water was 0, but I have added, suppose, 10 molecules of sugar to it, and now thus it has become solution, and the water potential of the solution, supposedly, it becomes -4. It becomes -4. So it has decreased from 0 to -4. And if I further add the molecules of sugar, suppose I have added 50 molecules of sugar, the water potential has reduced to -8. It has reduced further to -8. So it is getting reduced as you are increasing the solute concentration. So it has become the negative value. Now listen carefully. It was 0 before, but when I have added the sugar molecule, it has reduced to -4. So by what amount? By what amount the water potential has reduced? By what amount the water potential has reduced? 4. But it is necessary to apply minus in front because it is the minus will show that it is decreasing. So what will I say? That by what amount the water potential is getting reduced as soon as I add sugar molecules. So I will say by -4. And this is solute potential of the solution. And this is the solute potential. So now I am explaining another term to you, and that is solute potential, or psi S. See, the amount by which water potential decreases on addition of solute particles to water is called as solute potential. Clear? So I have added sugar molecules. The water potential of the solution will decrease. It has become, suppose, -4. Suppose the water potential has become -4. So obviously now the water potential of the solution becomes equal to the solute potential. Because when you add sugar, the amount by which the water potential gets reduced is solute potential. So psi W becomes equal to psi S. But under what conditions? Under the conditions of same standard temperature and standard pressure. You are not applying any external pressure to the solution. When you are not applying any external pressure to the solution, under the condition of standard pressure and standard temperature, the amount by which the water potential gets reduced on addition of the solute particles, that is the solute potential. That is, water potential and solute potential are the same values in this case. So psi W is equal to psi S. Both are -4 in this case. Are you getting the point? So I will say that both water potential and solute potential are inversely proportional to solute concentration. It is very common that the students think that water potential decreases when we add solute particle, But solute potential increases when we add a solute particle. It is very common question students ask, ma'am, if we increase the solute concentration, so solute potential will increase. No, both water potential and solute potential are inversely proportional, because when you are adding sugar to the pure water, first the solute potential or the water potential decrease to -4, and when you will add more sugar to it, it will become -8. So which is greater, -8 or -4? Obviously -4. So as you are adding the sugar molecules, both the water potential and the solute potential are getting decreased. So both of them are inversely proportional to solute concentration. For pure water, both psi W and psi S are 0. And for solution, both of them are negative values. Remember this. So they are a team. Psi W and psi S are a same team. They increase together, they decrease together. They are 0 for pure, both of them are 0 for pure water, and both of them are negative values for a solution. I hope this is clear to you. So you're clear with water potential, you're clear with solute potential. And yes, one more important thing, that solute potential is also called as osmotic potential. Very important. Solute potential is also called as osmotic potential, not osmotic pressure. Do not get confused with osmotic pressure and osmotic potential. These two are different things. Osmotic pressure, entirely different thing. Osmotic potential, entirely different thing. So another name of solute potential is osmotic potential. Okay, clear? Yes. Now, entry of another hero into the picture, and that is pressure potential, or psi P. Up till now, I have not applied any external pressure to the solution, but now I am applying it. So this is a solution, not pure water, solution. This is a solution, and I am applying external pressure to it. So when I will apply external pressure to the solution, the external pressure will try to increase the kinetic energy of the water molecules. So the external pressure will try to increase the water potential, right? So when I will apply the external pressure to the solution, the amount by which the external pressure tries to increase the water potential is called as pressure potential. Right? Sometimes this is also called as positive hydrostatic pressure or turgor pressure. Right? So this is written over here that the amount by which the kinetic energy of water molecule tend to increase when there is application of external pressure, that is pressure potential. So psi W and psi S was a negative value. It was a negative value, or it is 0 for pure water. But water potential is generally a positive value, right? So beta, pressure potential is a positive value, but there are two important cases. There are two important cases where the pressure potential is a negative value. In case of xylem vessels, psi P is negative. Why? Because in xylem vessels, there is negative pressure created by the transpirational pull. The pressure is maintained negative in it. In plasmolyzed cell, it is a cell from which all the water has come out and the protoplast is shrunken now. So there is nothing, no pressure applying outside the cell, is applied out towards the outer wall of the cell. So the psi P is negative. These two are the important cases. Inside xylem vessels and in the plasmolyzed cell. What is a plasmolyzed cell? I'll tell you in the later part. For both of them, psi P is negative. Otherwise, it is generally a positive value. So now I have something very important in front of me. Pure water. Water potential of pure water is 0 when no external pressure is applied. Some students ask, but ma'am, when we will apply some external pressure to the pure water, will the value will become positive? Yes, it can. It can. But what we are taking here is a standard value under standard conditions, right? If you apply external pressure to pure water, it may become positive. But right here, right now, we are trying to explain some standard condition we are taking as a reference value. So here, water potential of pure water is 0 under the standard conditions. Then we have made this water as solution by adding sugar to it. So water potential of solution become equal to solute potential when we are not applying any external pressure. But when we are applying external pressure, the water potential of the solution becomes equal to psi S plus psi P. Okay, so here these are three formulas. Look at them carefully. Pure water, 0, no external pressure. Psi W of solution is equal to psi S, no external pressure. Psi W of solution equal to psi S plus psi P. And there is one more important thing, that if I talk about the water movement occurs from high concentration to, from region of high water potential towards the region of low water potential. Water always moves from high water potential to low water potential. Remember this thing, this is very important. Okay, let's move. Now let's do a question. What kind of question comes from this kind of portion? You have three systems, A, B and C. Here it is given that psi S is equals to -8 and psi P is equal to 4. Here psi S is equal to -10 and psi P is equal to 6. Not 6, let's take it as 8. Psi S -12, psi P 6. You have to predict the direction of movement. So what we'll have to do here? We'll have to calculate the water potential. We know formula is psi S plus psi P. So -8 plus 4. Water potential will be -4. -10 plus 8. Water potential will be -2. -12 plus 6. Water potential will be -6. Clear? I have just told you that water will move from high water potential to low water potential. So, which has the highest water potential here? -2 is the highest, right? So water will always go from it because it has highest water potential. So it will give water both to B and to A. Then out of A and B, which one is greater? -4. So A will give water to B. That means B will not give water, it will only take water from both A and C because it has the lowest water potential. A will take water from C but will give it to B. Clear? So this would be the direction of movement of water. Okay? So remember this point. Now let's move on to another important case, that is special case of diffusion called as osmosis. How it is a special case of diffusion? Because here the movement again occurs from high concentration to low concentration of water, but it requires a special membrane, that is semi-permeable membrane. So because osmosis is also a special case of diffusion, it is passive in nature. But what is special here, that if I separate two solutions, one has the concentration 5 molar and one has the concentration 10 molar. And I have separated them with the help of semi-permeable membrane. Then obviously beta, this is dilute solution, so it will have more water concentration. This is concentrated solution, so water will be less. So what will be the direction of, and yes, one more important thing, that during osmosis, there is only movement of water molecules, that is solvent. There is never the movement of solute particles. Sugar or salt will never move across semi-permeable membrane. Only water will move. So remember, during osmosis, there is only movement of solvent particles or water, not solute. So here the water is more, here the water is less. So water will move from here to here, cross the semi-permeable membrane, and this is called as osmosis. In case of a living cell, if I talk about a living cell, the semi-permeable membrane or the selectively permeable membrane are two: plasma membrane and the membrane of the vacuole, that is tonoplast. In a plant cell, water has to cross two semi-permeable or selectively permeable membrane, plasma membrane and tonoplast. Clear? Okay. So, what can be the definition of osmosis? Read carefully. Movement of solvent particles, solvent, not solute, from less concentrated solution, 5 molar solution, in which there will be more concentration of water and high water potential, right? In the 5 molar solution, in this solution, the concentration of water was more, so water potential was also more. Towards high concentrated solution, and the high concentrated solution will have less concentration of O2, H2O, sorry. So it will have less water potential. Through semi-permeable membrane is osmosis, right? So movement of water from less concentrated solution towards more concentrated solution is osmosis, right? So water is moving from its own high concentration to its own low concentration here. Now, osmosis, remember this beta, thus osmosis is the only case of diffusion which depends on concentration gradient as well as pressure gradient. Otherwise, diffusion always depends only and only on concentration gradient, but it depends both on concentration plus pressure gradient. This question comes directly. This is very important. Then, these are the lines of NCERT, very important. Inside a plant cell, there is a vacuole which covers the 90% space of the plant cell. Inside the vacuole, most of the materials of the plant cell is stored inside the vacuole. So, most of the osmotic potential of a plant cell is decided by the vacuolar sap. The sap which is present inside the vacuole decides most of the osmotic potential of the cell. And when anything has to go into the plant cell, it has to cross two membranes. First, the plasma membrane, and second, the tonoplast. Clear? So these are very important lines. The question can come directly. Most of the osmotic potential of a plant cell is decided by, so your answer will be vacuolar sap. Clear? Now, osmotic pressure. When the water molecules are getting transferred from the semi-permeable membrane, then the minimum pressure I apply here to just stop the movement of particles, to just stop the movement of water molecules, that is osmotic pressure. The minimum pressure applied to just stop the movement of solvent particles is the osmotic pressure. And osmotic pressure, as you read in chemistry, it is equal to ICRT. C is the concentration here. So, what does it mean? That osmotic pressure is directly proportional to solute concentration. The more will be the solute concentration, the more will be the osmotic pressure of the solution. Right? This is important. And there is a very interesting relationship between osmotic pressure and osmotic potential. Pi is equal to minus psi s. If someone has given that the osmotic potential of the cell is minus 10, what will be its osmotic pressure? Simply 10. So this is a very important relationship between osmotic pressure and osmotic potential. Remember this one. Clear? Yeah. Now, before going to that, you have to remember one thing, beta, that the movement of water in context to pi occurs from low pi to high pi. Low pi to high pi. In context to water, what I have just told you, in context to water, that Water, in context to water potential, water moves from high water potential to low water potential. But in context to osmotic pressure, water moves from low pi to high pi. How? The solution which has low osmotic pressure means the solute concentration is also low. If the solute concentration is low, that means water potential is high. So water will move from high water potential to low water potential. That means from low osmotic pressure to high osmotic pressure. Clear? Remember this one. Water moves from low osmotic pressure to high osmotic pressure, but in context to water potential, it moves from high water potential to low water potential. Okay. Now let's define another important thing, that is DPD. What is DPD? Before going to that, let's understand what is diffusion pressure. When during diffusion, the water molecules move, when the water molecules move during diffusion, they move with a pressure, and that is called as diffusion pressure. So obviously, when the water molecules are pure, they will move fast. So they will move with high pressure. So the diffusion pressure will be high. But when you add sugar molecules to it, they will move slowly. So they will move with less pressure. So the diffusion pressure will be less. So when you add sugar to the pure water, the pressure with which the water molecules will move will decrease. Suppose when the water was pure, the diffusion pressure of water molecules was 10 atm. But when you have added sugar to it, it has reduced to 5, or suppose say 4. So by what amount it has decreased? 6. 10 minus 4, 6. So what is DPD? DPD is diffusion pressure deficit. The amount by which the diffusion pressure of water molecules get decreased if you add solute particles to it, that is diffusion pressure deficit. And DPD tells you that a cell has deficit of water, that the cell is in need of water. The more the value of DPD of any cell, because DPD is deficit, it is representing deficiency, right? So DPD, diffusion pressure of pure water minus diffusion pressure of solution, that would be diffusion pressure deficit. Another formula for DPD is osmotic pressure minus turgor pressure. What is turgor pressure? When a cell is turgid, when water enters into the cell, a cell becomes turgid, right? So the protoplasm exerts a pressure towards outside the cell, that is turgor pressure. So osmotic pressure minus turgor pressure gives you the DPD value of a cell, right? DPD means deficiency, right? Wherever there is deficiency, that means things will not go from there, things will come to there. And wherever there is no deficiency, things will go from there. So if the DPD is high, that means deficiency is high. That means water will come to it. And if the DPD is low, that means the deficiency is low. That means water will go from there. So in context to DPD also, the movement of water occurs from low DPD to high DPD. Right? From low DPD to high DPD. Because low DPD means less deficiency of water. That means water will go from there. Okay? So here we'll discuss 3 cases. Case of fully flaccid cell. Fully flaccid cell means a cell which is, which is flaccid, which have very less amount of water in it. In a fully flaccid cell, the turgor pressure becomes 0 because there is no one inside to exert pressure outside. So turgor pressure is 0, so DPD becomes equal to OP. For fully turgid cell, a cell which is, maximally satisfied with water and does not need a drop of water. So there is no deficiency of water in it. So a fully turgid cell has DPD 0 because it has no deficiency of water in it. But in a plasmolyzed cell is a cell in, if we put a plant cell in a hypertonic solution, all the water goes out of the cell and the protoplast of the cell gets shrinked. So remember, the turgor pressure was negative, the psi p was negative for a plasmolysed cell. So the psi p or turgor pressure is negative. So OP minus TP becomes OP plus TP. So for a plasmolysed cell, DPD is equal to OP plus TP. See, so DPD is so high for a plasmolysed cell. Very, very high, right? That means a plasmolysed cell is dying to get water. So these are the three cases. Okay, now let's make a team. We have made two teams here, team A and team B. In team A, water potential and solute potential are there, and in team B, pi or DPD is there. Water potential and solute potential, both of them are inversely proportional to solute concentration, but pi and DPD both are directly proportional to solute concentration. Psi W and psi S, for pure water, both of them are 0, and for a solution, both of them are negative values. But osmotic pressure is generally a positive value. For a living cell, osmotic pressure is not 0, cannot be 0. DPD for a cell can be 0 or it can be a positive value. That means DPD and pi are positive values, but psi W and psi S are negative values. In context to psi W and psi S, water movement occurs from high concentration to low concentration. But in context to pi and DPD, water movement occurs from low to high, from high DPD, from low DPD to high DPD, from low pi to high pi. Here, from high water potential to low water potential. So we have made these two teams, right? So they will increase together, decrease together. They will increase together, decrease together. When these two will increase, these two will decrease. Clear? And there is one important relationship between DPD and psi W also. Like pi was equal to minus psi S. Likewise, DPD is equal to minus psi W. If water potential of a cell is minus 10, then DPD would be 10. Okay? Now, imbibition. Imbibition again is also a special case of diffusion. So there are 4 cases of diffusion: simple diffusion, facilitated diffusion, osmosis, imbibition. These all are cases of diffusion. These all are passive process, and they all depends on concentration gradient, except osmosis, because it depends both on concentration and pressure gradient. Imbibition, how it is a special case of diffusion? Because it needs a dry surface to occur. Now, what does a dry surface means? The surface of a seed. It occurs on the surface of a mature seed and the dry wooden surfaces. If the question comes that you have put a raisin in the water and it gets swelled up, it is imbibition. False. This is not imbibition. This is endo-osmosis. But if you put a seed into water and it gets swells up, this is imbibition. If you put a piece of wood into water and it gets swells up, this is imbibition. Clear? So imbibition occurs on dry surfaces like mature seed and dry wooden surfaces. Clear? So the surface on which imbibition occurs is called as the imbibent, and the molecule which gets absorbed on the surface is called as imbibate, and generally the imbibate is water. So water gets absorbed on the surface and then absorbed into the surface. And when the water molecules gets absorbed into the surface, the kinetic energy gets transformed into heat energy. So imbibition is an exothermic process in which heat energy is released. Okay, is this clear? Now, what are the conditions that are very important for imbibition? Obviously, a concentration gradient is very important. Without concentration gradient, imbibition will not occur. Affinity between imbibate and imbibent. Affinity means the surface on which imbibition occurs and the affinity between the water molecules. Affinity means they should love each other, they should have affinity for each other. That means this surface should be the kind which can easily allow the absorption of water on it. If the surface is made up of phycocolloids, the rate of phycocolloids like agar-agar, olgin obtained from the red algae, if the surface is made up of phycocolloids, then very high rate of imbibition, then pectin, then protein, then starch, and then finally cellulose, right? Then imbibition pressure. When the water molecules gets absorbed into the seed or wooden surface, then they exert a pressure outside, and that is called as the imbibition pressure or the matrix potential. If this pressure is restricted and you are not allowing it to escape, then this pressure can build up to 1000 atm, and this particular pressure, imbibition pressure, is used during seed germination. When water enters into the seed, it exerts a pressure with the help of which the seedling comes out and seed germination occurs. Initially, we used to break big boulders and mountains with the help of this imbibition pressure, because it can, if it is restricted, it can grow up to 1000 atm, right? We used to make cracks in the boulders and the mountains, and then we used to add some wooden pieces in those cracks and then add water to it. They used to get swell up, but the imbibition pressure, it used to restrict and grow up to 1000 atm, which resulted in the breakdown of boulders and mountains. Okay, so this is imbibition. Now I hope this much is understood. So what we have discussed up till here, the short distance transport and the various kind of diffusion, the simple diffusion, the facilitated diffusion. I hope this much is clear to you. So now let's discuss plasmolysis, but before going to that, let's understand the different types of solutions, right? First of all, let's go to an isotonic solution, right? See beta, this is the plant cell, this is the plant cell, and this is the solution inside the cell, that is called as cell sap. So when the concentration of outer solution is equal to the concentration of the solution present in the cell or cell sap, that is an isotonic solution. That means in this case, the water potential of the solution and the water potential inside the cell are similar. So there will be a condition of equilibrium, right? The amount of water going in will be equal to the amount of water going out. So the net movement of water will be zero. Net water movement will be zero. This is wrong. Net movement of water will be zero. And there is a condition of equilibrium. So when a plant cell is put in an isotonic solution, there will be no change in the volume of solution, in the volume of cell. In the volume of cell, there would be no change. So here, the water moving into the cell will be equal to water moving out of the cell. So no net movement of water. No net movement of water, and there will be condition of equilibrium. Then comes the hypotonic solution. In this condition, the concentration of outer solution is lesser than the concentration of cell sap. And if the outer solution has lesser concentration, that means the water potential of the outer solution will be greater. So water potential of outer solution is greater than the water potential of the cell sap. That means the pi, because pi and psi w are in the opposite team. So the pi of solution will be lesser than the pi of cell sap. And you know that water move from high water potential to low water potential, or low pi to high pi. So this outer solution has high water potential and low osmotic pressure. So water will move into the cell. Water will move into the cell. This is called as endoosmosis, and the volume of the cell will increase. So whenever you put a plant cell in hypotonic solution, the volume of the cell will increase. This is called as endoosmosis. The example of resin I just gave you. When you put a cell in hypertonic solution, in this case, the concentration of outer solution, the outer solution is greater than the concentration of the cell solution. So obviously, water potential of the cell is greater here. Water potential of the cell is greater. So water will move out of the cell. This is called as exoosmosis, and the volume of the cell will decrease in hypertonic solution, and this is the basis of plasmolysis, right? So when you put a plant cell in isotonic solution, there will be no change in the volume of cell. When you put a plant cell in hypotonic solution, endoosmosis will occur and the volume of cell will increase. When you put a cell in hypertonic solution, the volume of cell will decrease and exoosmosis will occur, and this is the basis of plasmolysis, right? So let's move on. What is plasmolysis actually? During plasmolysis, when you put a plant cell in hypertonic solution, when you put a plant cell in hypertonic solution, then the water will come out, right? Now, what will happen that when water will come out, this protoplast, what is protoplast? The living material of the cell, plasma membrane, cytoplasm. The protoplast will shrink. The protoplast will shrink. And this is called as plasmolysis. This is plasmolysis, right? So what will happen that the cell wall will have no effect on it because cell wall is already dead. But the living material, that is protoplast, the plasma membrane, the cytoplasm will shrink and form a ball-like structure at the center. So this is a plasmolyzed cell. This is a plasmolyzed cell. Clear? Now these are the lines of NCERT, very important. When you put a plant cell in hypertonic solution, water will come out of it, but the water comes first from cytoplasm. All the water which is present in the cytoplasm, it goes out, and then the water present in the vacuole, it goes out, and then the cell becomes plasmolyzed. So this is very important. During plasmolysis, the water first go out from cytoplasm and then from vacuole. And there is one more question which is very important. This is the shrunken protoplast. This is the shrunken protoplast. The protoplast has shrinked and formed a ball-like structure, and this is a cell wall. There is no effect on the cell wall because cell wall is already a dead layer. So there is a space between this shrunken protoplast and the cell wall, and this space is filled with hypertonic solution, because cell wall is a fully permeable wall. So it is present in hypertonic solution, and the hypertonic solution would get filled in this space. This is very important. In a plasmolyzed cell, the space between shrunken protoplast and the cell wall is filled up with hypertonic solution. Okay? Now this is the diagram of NCERT, important. Here you can see beta that this is a plasmolyzed cell. Water is going out of the cell. Water is going out of the cell. And the protoplast, this is the plasma membrane. The plasma membrane is getting detached from the cell wall, and the protoplast is getting shrinked towards the center. This is a flaccid cell. Here you can see that the water going outside the cell is equal to water going into the cell. So this is a state of equilibrium. Here you can say that the water is coming into the cell. So obviously This is a cell which is present in hypotonic solution. This is a cell which is present in isotonic solution. And this is the cell which is present in hypertonic solution. So obviously, the cell, when it is put in hypertonic solution, it is called as plasmolized. When it is present in isotonic solution, it is called as flaccid. And when it is present in hypotonic solution, it is called as turgid. Okay? Now deplasmolysis. Up to a limit, the process of plasmolysis is reversible. That means if you put a plasmolized cell into distilled water or hypotonic solution or water, then the water will get filled up into the cell and it will again come back to life, because the shape is not, it's not retorted, because the cell wall is not affected by it. So up to a limit, the process of plasmolysis is reversible when you put the plasmolysed cell in water or in hypotonic solution. Okay. Now yes, now let's move on to the long distance transport. When I talk about the long distance transport, up till here we have discussed what is short distance transport, what is simple diffusion, facilitated diffusion, osmosis, imbibition, types of solution, plasmolysis, right? Now comes the second part of this chapter, and that is long distance transport, also called as translocation. So here diffusion fails. Diffusion is a very slow process, and the rate of diffusion is 50 micrometers per 2.5 seconds. See, in 2.5 seconds, the materials will travel a distance of only 50 micrometer, 10 raised to the power minus 6 meter. The leaf will wait and die when the water and minerals will come. So a plant cannot depend upon diffusion for long distance transport. So here, very important things come into picture, and that is mass flow or bulk flow. For long distance transport, we talk about mass flow, and this mass flow depends on pressure gradient. Remember, this is very important. Up till here we have talked about that diffusion depends only on concentration gradient. Exception was osmosis. Osmosis was dependent on both concentration and pressure gradient, but mass flow is independent. Listen carefully. Mass flow is independent of concentration gradient and depends only on pressure gradient. If there is difference between pressure between 2 points, mass flow will occur. How can you take the example? Normally, the origin of Ganga river is in Himalayas, right? Over there is a melting of glacier, and the water, large amount of water gets accumulated and the pressure becomes high. But in the plains, the pressure is low. So you never, take Ganga and pull it towards the plain. It comes towards the plain by itself, because there in the origin point, the pressure of water is high, and in the plains, the pressure of water is low. So the water comes by itself and takes whatever comes in its way. This is mass flow. Likewise, in plants, we create difference between 2 points, pressure difference, and the water and the things, they get dissolved in water and gets transported in this flow called as mass flow. Okay? So we have two kind of forces which are helpful in mass flow in the plants. One is the root pressure or the positive hydrostatic pressure, and the another force is the transpirational pull or the negative hydrostatic pressure. What is the example of the positive hydrostatic pressure? How it is positive? You can take the example of, when you have to, give water to the garden, then what you will do, you will connect the pipe to the tap. You will open the tap, and from one point you will increase the pressure and water will flow into the pipe by itself. This is positive pressure, right? But when you put a straw into a cold drink and take a sip, You created negative pressure in the straw, and there's high pressure in the bottle of cold drink. The cold drink will rise into the cold straw, and you will, enjoy the cold drink. So this is negative pressure. This is mass flow, but with the help of a negative force. And when you've opened a tap, this is also mass flow, but with the help of a positive force. So in the plants, root pressure is a positive hydrostatic pressure, and transpirational pull is like a straw thing. So it is negative hydrostatic pressure, right? These two forces together are helpful in the long distance transport, that is mass flow. Let's discuss them in detail now. First of all, let's understand how plants absorb water from the soil. Okay, so this is a root, this is the epidermis of the root, and this is the root hair. This root hair is in direct contact of the soil. Okay, water will cross the membrane of the root hair with the help of diffusion, because concentration of water is always high in soil as compared to the root. So the absorption of water by root hair is done by diffusion. That means it is passive, no expenditure of energy. And when you want to be specific, it is a diffusion, osmosis. So this is very important. Direct question come. How do plants absorb water? Your answer will be by diffusion. And if you want to be more specific, by osmosis. It's a special diffusion, osmosis, and it is passive, no expenditure of energy. Clear? Direct question comes. Then ultimately the root hair has absorbed the water from the soil, but it has to ultimately it has to fill up into the xylem, and from the xylem of the root it has to be transported up till to the xylem of the leaf, right? So the root hair has absorbed the water from the soil by diffusion or osmosis, but now the water has to cross many layers to ultimately get to the xylem. Xylem is present here, right? So this we will study in detail in anatomy. What is the structure of a root? A root has epidermis, then inside epidermis it has cortex, then endodermis, then pericycle, and then finally xylem. So root hair has absorbed water from the soil, but now the water has to cross many layers to ultimately reach up to the xylem. So what kind of movement is shown by the water when it is crossing these different layers? So water shows two kinds of movement, epoplast movement and symplast movement, when it will cross these different layers of the root. What kind of movement is epoplast? During epoplastic movement, the water crosses only the dead space. The water do not cross the plasma membrane. That means if water will move from intercellular spaces to intercellular spaces, or from cell wall to cell wall, because cell wall is also dead. So movement of water from cell wall to cell wall or from intercellular spaces, that means it is moving through epoplast, and epoplast is the dead space. It is not crossing the plasma membrane. But when the water crosses the plasma membrane and enter into the living space of the cell, that is the symplastic movement. Okay, so here you can see that water is moving through intercellular spaces or from cell wall to cell wall. But here the water enters into the plasma membrane and enters into the living space of the cell. Obviously the epoplastic pathway is faster because it is moving through dead space, but the symplastic pathway is a bit slow. Majority of pathway of water is epoplastic. Majority of it. How? Let's understand. When the water has to cross cortex, the first, after absorbed by the epidermis, it will go to cortex, right? The cortex has large intercellular spaces. So to cross the cortex, the water will take apoplastic pathway, dead space. But then comes the endodermis. To cross endodermis, water takes the help of symplastic pathway. Then pericycle, again majorly apoplastic. So most of the pathway water takes is apoplastic, but to cross endodermis, it takes the help of symplast. Why? I'll tell you that. So majority of pathway is done with the help of apoplast, but to cross the endodermis, symplast is taken. Why? I will just tell you. Now there is some notion, students think that because the water will cross the plasma membrane in symplastic pathway, so it will be an active process. No, do not think like that. Both the processes, these are the lines of NCERT, both the processes occurs down the gradient. That means both of them are passive in nature. But yes, because in symplastic pathway, the living space of the cell is involved, so symplastic pathway can be helped by cytoplasmic streaming. Symplastic pathway can be helped by cytoplasmic streaming, and you know cytoplasmic streaming is active, right? So generally apoplastic pathway and symplastic pathway both are passive, occurs down the gradient, but in case of symplastic pathway it can be helped by the cytoplasmic streaming, right? Now in the cortex layer there are large intercellular spaces, so apoplastic pathway. To cross endodermis, symplastic pathway. To cross pericycle, it may be symplastic, but majorly it is apoplastic. Xylem, you know that xylem is a dead tissue. Inside xylem there is nothing. So when the water will be moving through xylem, that means it is moving through a dead space. And what kind of movement is that? Apoplastic. So imagine there is root. The outermost layer of the root has absorbed the water from the soil passively by diffusion. Then the water will cross the cortex, apoplastic pathway, endodermis, symplastic pathway, pericycle, mostly apoplastic. Then it is filled in xylem. From xylem, it will move to the xylem of stem, then to the xylem of leaf. And the whole xylem is the dead tube. So when the water is moving through the xylem, it is actually apoplastic space, dead space. So majority of the movement of water is apoplastic. Okay? Then why to cross endodermis, the movement has to be symplastic and not apoplastic? Because on the wall of endodermis, there is a special covering of a special material, that is suberin. And suberin is a waxy compound which is impermeable to water. And these depositions of suberin are called as Casparian strips. So these Casparian strips are present on endodermis, which do not allow the easy entry of water into the endodermis. So but there are some portions, there are very few portions where this suberin is not there. Very few portions where the suberin is not there. So water afford to take entry from those points, and then it crosses the plasma membrane, that with the help of plasmodesmata, remember the bridge-like structure connecting the two cells. So suberin is deposited on the cells of endodermis, but there are few points where it is absent. Water afford to take the entry, then it crosses the plasma membrane. With the help of plasmodesmata, it moves through the living space of the cell and then comes out. So this is why to cross endodermis, water has to take the help of symplastic pathway and not the apoplastic pathway. Clear? Now this is the diagram of NCERT. You can see that Up till here, the movement was through cell wall, but here at this point when it has to cross the endodermis, the water has entered into the cell and the water will come out through plasmodesmata. Okay, so cross the endodermis, symplastic pathway is taken. Now we have seen that the outermost layer of the root, that is epidermis, absorb the water. Now water has moved across several layers with the help of epoplast or symplast, and now the water is filled in the xylem. But from the xylem of root, it has to move up against the gravity, and this is called as ascent of sap. Water and the minerals present in the xylem has to move against the gravity. This is called as ascent of sap, which is done with the help of root pressure and with the help of transpirational pull. First, understand what is root pressure, right? Let's understand this and what are the questions that have come from this portion. Okay, now what is root pressure and how root pressure in the root gets generated? Understand this. This is a root, right? This is soil. First of all, mineral ions are taken up from the soil by the root. These mineral ions will get deposited into the xylem. Mineral ions are taken from the soil and these mineral ions will get deposited into the xylem. As soon as the mineral ions will accumulate into the xylem, minerals are solutes. What they will do? The mineral ions will reduce the water potential of xylem. Because of the accumulation of mineral ions in the xylem of root, the water potential of xylem gets highly reduced, and the water from the soil goes into the xylem with a pressure, and that pressure is root pressure. Okay, the water potential of water in soil is very high, and due to accumulation of mineral ions in the xylem, the water potential of xylem gets reduced, and due to this, the water gets filled up into the xylem of root with a pressure, and that pressure is root pressure. And this is a positive hydrostatic pressure. The another name for root pressure is positive hydrostatic pressure. Now the question which has come in 2015, that root pressure develops actively or passively. What will your answer? What will be your answer? Root pressure develops actively or passively? So your answer will be root pressure develops actively. How? Because to absorb these mineral ions from the soil, energy has to be given. Energy is used. So first of all, the root will take the mineral ions from the soil. For that, it will expend energy. Then these mineral ions will be accumulated in the xylem. The water potential of the xylem will get reduced, and the water from soil will fill up into the xylem with the pressure, that is root pressure. So to develop root pressure, you had to absorb, the root had to absorb the mineral ions, and that is active in nature. So development of root pressure is active. Okay, it's active. This is important. Now the water potential, now what yes, so this is what I have just told you. It is written that the water potential of the soil is much higher than the water potential inside the xylem because of the accumulation of mineral ions, and then the water will gets filled up. Clear, beta? Now root pressure, when the root pressure is maximum in a plant, when the rate of absorption of water is much higher than the rate of transpiration. Imagine you have opened a tap and you have tied the pipe around it, and when will the pressure be maximum? When the water is flowing through the pipe, or when you have put a thumb on the other end of the pipe. When you have opened the tap and put the thumb on the other end of the pipe, the pressure in the pipe will be very high. But if the water is just flowing out of the pipe, the pressure in the pipe will not be high, right? So if the soil is absorbing water and simultaneously the water is evaporated through transpiration, so how will the root pressure will develop? So root pressure is high during evening and the early morning hours when there is negligible amount of transpiration, because the water is not going out, only absorption is happening. So there will be pressure in the xylem vessels. Okay, so in the morning or evening hours when transpiration is almost absent, then the root pressure is high. When the rate of transpiration is much higher than the rate of when the rate of absorption is much higher than the rate of transpiration, then the root pressure is maximum. Clear? So when does it happen? During early morning and late evening. Now what are the processes that occurs in the plant with the help of root pressure? Those are guttation and bleeding. Bleeding is simply, what is bleeding? When there is a cut in the plant, there is an injury in the plant, then the sap comes out with a, sap comes out like this, exudation of sap. This is bleeding, and that is also occurs with the help of root pressure, but the main process is guttation. So understood what is root pressure? When the root absorbs mineral ions actively from the soil, the mineral ions gets accumulated into the xylem vessel, water potential of xylem vessel get reduced, water fills into the xylem with the pressure, root pressure. Root pressure is maximum during morning and evening hours because then the transpiration is almost absent. Clear? Now the root pressure is not very beneficial with the plants which have a great height, because root pressure has to move against the gravity. So after a height, it will be like this. Oh, I'm tired. So because root pressure has to work against the gravity, so for very tall plants it is of no use. It is beneficial only for small heighted plants. And moreover, root pressure is not the major force in the plant which helps in the ascent of sap. It is just a supporting force. The main force is transpirational pull. Okay. Now let's understand guttation. Guttation is the exudation of water in liquid state, right? In transpiration, the water comes out of stomata in gaseous state. But in guttation, there is water exudation. The water comes out in the liquid state and with impurities, right? And in guttation, the water comes out through a special pores, and those special pores are called as hydrothoids. And these hydrothoids are present in the margin of leaves. Margin, not on the surface, on the margin of the leaves. But these hydrothoids are not present in all the plants. They are present only in some herbaceous plants. Hydrothoids are present only in some herbaceous plants like grasses and tomato. This is very important. This process of guttation is not found in every plant. This is found in some herbaceous plants like grasses and tomato, in which hydrothoids are present in the margin of leaf. And due to the root pressure during the evening and early morning hours, the water through these hydrothoids come out, and that is called as guttation. So you can see guttation only during early morning hours or evening hours, not during day. Now comes the major force behind the ascent of sap. How the water actually goes up the plant against the gravity. And the most accepted theory is cohesion, tension, transpirational pull theory, given by Dixon and Joly. This theory says that many forces together helps the water to get to the leaves against the gravity. The very first force is the cohesive force. Cohesive force is the attractive force between the water molecules. Then comes the adhesive force, attractive force between water molecules and the xylem wall, right? So when the water molecules are moving against the gravity, they are holding hands of each other, so that if they will fall together. So this, they are holding hand together. This is cohesive force. And they are moving like this. They are attached to the xylem wall and they are moving like this, right? So if they will fall, they will slide, they will not fall abruptly, right? So they are holding hands of each other and they are, moving because of the adhesive force with the xylem. Okay. Then surface tension. When the water in the xylem moves in the liquid states, the surface tension is very high. The inward attractive force is very high, which helps the cohesive force to develop high. You must have read surface tension in physics also, right? Then comes capillarity action. The diameter of the xylem vessels is narrow, and because of the narrow diameter, capillarity action is there and it helps in the upward movement of water. And the most important force is the transpirational pull. That is the pulling force. The pulling force, which is also called as negative hydrostatic pressure. Root pressure is positive hydrostatic pressure, but transpirational pull is negative hydrostatic pressure. So these forces together, cohesive force, adhesive force, surface tension, capillarity, transpirational pull, all together, they are helpful in the ascent of sap, movement of water and minerals against the gravity. And here the helping force is root pressure. Okay? Clear? Now what is transpirational pull? Because of these forces, because of the various forces, the water column has very high tensile strength. The water and minerals moving in the xylem, they have very high tensile strength. That means you cannot easily break the water column in the xylem vessel. Now, how the transpirational pull develops? During day, through stomata, the water gets evaporated. Okay? Because of that, the water potential of mesophyll cells get reduced. So mesophyll cell will take water from the xylem of leaf, which will reduce the water potential of xylem of leaf. The xylem of leaf will take water from the stem of xylem, from the xylem of stem. This will reduce the water potential of stem of xylem. Then the xylem of stem will take water from the xylem of root. This will reduce the water potential of root. So everybody is reducing water potential of each other, and that is why ultimately the water potential at root is very low and it will take water from the soil. So in the whole column, in the whole xylem tube, the negative pressure is created because of the transpiration. So there is a pull-like force which is generated, that is transpirational pull. That is the major force in the ascent of sap. Now what is transpiration? Transpiration is evaporation of water through stomata in the gaseous state. In guttation it was liquid state, but here it is gaseous state. And the water which comes out of stomata is pure water, right? Transpiration generally occurs through stomata present in the leaves or sometimes in the stem. It can also occur through bark, right? It can also occur through lenticels present in the woody stem, and it can also occur through the cuticle in the leaf. There are cracks in the cuticle which can, from where the water can evaporate. But this is not very important from the NEET point of view. Majorly, the transpiration occurs through stomata. Now, you know that for transpiration to occur, you need stomatal apparatus, and the detail of this will be dealing in anatomy. But here you can see, beta, that the very important thing in transpiration is stomatal apparatus. Inside there you can see guard cells, right? These are the guard cells, At the center, there is a pore called as the stomatal pore. These are the guard cells and these are the subsidiary cells, the epidermal cells surrounding the guard cell. Now here you can see these lines. These lines are the radially arranged cellulosic microfibrils. This is very important. Sometimes in the question it is written that guard cell have longitudinally arranged microfibrils. No, these microfibrils are arranged radially in the guard cell and they help in contraction and relaxation, right? One more important thing, that this is the inner wall of the guard cell which is present towards the pore, and this is the outer wall of the guard cell. So what is important here? These are the important points, that the guard cells have radially arranged cellulose microfibrils. Then the inner wall of the guard cell is thick and elastic. Lines of NCERT, very important. The outer wall of the guard cell is thin, and in the epidermal cells there is no chloroplast. In the epidermis, chloroplast is absent, but guard cells are the only epidermal cells which have chloroplast, right? So this is the stomatal apparatus which help in transpiration. What you have to remember about it? What is the nature of inner wall of the guard cell, outer wall of the guard cell? Radially arranged microfibrils. Okay? Clear up till here? See beta, is it clear up till here? Yes beta. Now let's see how these guard cells, they regulate the opening and closing of stomata. And for that we'll have to deal with the potassium ion theory or the Levitt theory. Let's see what it is. So this here beta is a guard cell, right? This is a guard cell, and surrounding the guard cell is the epidermal cell, that is the subsidiary cell. This is the vacuole of the guard cell. Okay, what happens during day, that the starch present in the guard cell, because you know guard cell has chloroplast, so it will form food and the starch will be present over there. The starch will undergo a hydrolysis, it will form glucose, and then it will undergo EMP. What is EMP? The Embden-Meyerhof pathway, the glycolysis. So what will be formed? PEP, that is phosphoenol pyruvate. This phosphoenol pyruvate will combine with CO2 in the presence of the enzyme pepcase, and then oxaloacetic acid will be formed. Then from this oxaloacetic acid, malic acid will be formed. This malic acid will break into its ions, that is hydrogen ions and the malate ions. Then in the membrane of the guard cell, we have hydrogen potassium ion exchange pumps. Hydrogen potassium ion exchange pumps. What they do, they throw the protons or the hydrogen ions outside the cell and take the potassium ion into the cell. Along with them, chloride ions also come into the cell. So this question is very important, that during opening of stomata, what goes out of the guard cell? Hydrogen ions. And what comes into the guard cell? Potassium ions and chloride ions. So during opening of stomata, there is efflux of hydrogen ions, exit, and influx of potassium and chloride ions. These potassium ions combine with the malate ions and form potassium malate. This potassium malates get accumulated into the vacuole of guard cell. And you know potassium malate is a solute. You know that accumulation of solute decreases the water potential of the cell. So because of the accumulation of potassium malate in the guard cell, the water potential of guard cell decreases, due to which water from the subsidiary cell goes into the guard cell. Because of this, the guard cell become turgid. And when the guard cell will become turgid, the pore will get open and stomata will open. Clear? So this is important. So what happens during day? During opening of stomata, guard cells become turgid and stomata open. And what happens during opening of stomata? There is efflux of hydrogen and influx of potassium and chloride ions. Okay, so this is the basis behind the opening of stomata. Now transpiration is very important to plants, but it is evil also, because the water is getting so much, is taking so much effort in absorbing the water, and transpiration is just, you know, evaporating it. So it is evil, but it is necessary. Why? Because it creates, because of transpiration, transpirational pull is created. Because of transpiration, there is supply of water for photosynthesis in the leaves, right? Because of transpirational pull, the minerals gets transported to the various parts of the plant. Because of transpirational pull, transpiration occurs, and this transpiration reduces the temperature of the leaf by 10 to 15 degrees and generates a cooling effect for the leaf. And because of this transpirational pull, the different parts of the plant gets water, so the plant cells, they get the water, they become turgid, and their shape is maintained. So these all are the benefits of transpiration, right? Given in NCERT, important. Now there is one more thing which is given over here, transpiration ratio, in these lines, in the lines below. What is transpiration ratio? That what amount of water is lost when the CO2 is fixed during photosynthesis. So in these lines it is mentioned that in the C4 plants, this we will, you know, deal with the chapter photosynthesis, what are C4 plants and what are C3 plants. But in C4 plants, when photosynthesis happens, to fix one CO2 molecule, the loss of water is less. But in C3 plants, during photosynthesis, to fix CO2, the loss of water is more. So this is given in NCERT here, that the loss of water in C4 plants is just half of C3 plants. That means C4 plants are more efficient in, you know, in regulating the amount of loss of water. Clear? Now, what are the factors which affects the rate of transpiration? The factors can be external factors. Light. The more the light, the more would be the rate of transpiration. Humidity, inversely proportional. If the humidity is more, transpiration will be less. Temperature, directly proportional. Wind speed, if the wind speed is more, rate of transpiration would be more. Available soil water, if the water is available more, rate of transpiration will be more. Then internal factors, the factors of plant. The structure of leaf. If the number of stomata will be more in leaf, there will be more, you know, transpiration. But some leaves, they reduce the rate of transpiration because they have sunken stomata. Sunken stomata, the stomata are not exposed, but they are, you know, embedded into the epidermis. And thick cuticle. Cuticle is a waxy covering on the leaf. So some leaves have thick cuticle and the sunken stomata, like in C4 plants and in desert plants, and this reduces the loss of water. Number and distribution of stomata. If both the surface of leaf will have stomata, then rate of transpiration will be more, as in monocot leaf. But in dicot leaf, only the lower epidermis have stomata, the upper epidermis nearly absent, stomata nearly absent. Then plant water status. If plant has more available water in it, inside it, the rate of transpiration would be more. Canopy structure. If the canopy structure, if the canopy of the tree is high, then the rate of transpiration would be more. So these are the internal factors of the plant which affects the rate of photosynthesis. Transpiration. Okay, now let's move on to uptake of mineral ions. Up till now we have discussed that how plant take the water from the soil and how it is, you know, transported in the different parts of the plant. But how the plant take minerals from the soil? This is very important. Remember, I told you that the root hair absorb water from the soil passively, but the root hair absorb minerals from the soil actively. Actively. Now the question comes, what is the reason that mineral ions are absorbed actively? Two important reasons. First, the mineral ions in the soil are present in the charged form. And the charged form of ions is not easily acceptable by the plasma membrane. Second reason, the concentration of the mineral ions in the soil is always less than the concentration of the mineral ions inside the root. That means if the mineral ions have to move from soil into the root, it has to be done from low to high concentration, from low to high, that is against the gradient, and that is active. So remember, two reasons why the uptake of mineral ions is active. First, the mineral ions are present in the charged form, and second, the concentration of mineral ions in root hair is more than the concentration of ions in the soil, right? But very few of the mineral ions are absorbed passively also. So this is the line of NCERT, that the absorption of mineral ions by the root hair from the soil is both passive and active, but mostly it is active. Clear? Now, translocation of mineral ions. How mineral ions gets translocated from root to different parts of the plant? Obviously, it is done along with the help of water. It gets dissolved in the water and they get translocated with the help of water, with the help of mass flow. Now, like water was, the movement of water was, restricted, was checked at the endodermal point. Likewise, the movement of mineral ions is also checked at this point. So endodermis is a checkpoint. Endodermis is a checkpoint which regulates the movement of mineral ions. Suppose some harmful mineral is coming from the soil, suppose uranium. So this endodermis will not allow uranium to go into the xylem. It will stop it here and here and here. It will allow only useful mineral ions to go to the xylem. This means this endodermis, the endodermis, because it has suberin in it, this endodermis is a very special, layer in the root which regulates the movement of mineral ions and maintains the unidirectional flow. If the mineral has got into the xylem, it will go towards the different parts of the plant, right? So this is a important checkpoint in the movement of water as well as in the movement of mineral ions. Clear? This layer is very, very important. Now, how minerals gets translocated via xylem? Obviously, minerals get filled up in the xylem. Remember, minerals get filled up in the xylem. Because of that, the water potential of xylem gets reduced and the water from the soil gets filled up into the xylem. So there is a mixture of water and minerals here now into the xylem, and with the help of transpirational pull, this mixture of water and minerals is translocated towards different parts of the plant, right? So the sink for minerals, sink means the parts will need the minerals, which need the minerals. Source is root from where the minerals are going upwards, but the sink are the leaves which need mineral elements, right? Okay. Now let's see what happens when the water and minerals, they get translocated via xylem. So see here, this is a leaf. See, this is a leaf, right? And these are, these are the veins. And these are the veins in the leaf. Remember, beta, in the leaf, wherever there are veins, vascular bundles are present. You know what are veins in a leaf? Wherever there are veins, there are vascular bundles. So this is a leaf. These are the veins of leaf, and this is the xylem, because wherever there is vein, there is xylem or phloem. So this xylem is carrying the water and minerals from where? From root, right? Now this leaf is the sink. The cells of the leaf are called as the mesophyll cells. What are the mesophyll cells? They are the photosynthetic cell of the leaf, right? They need the mineral elements for their proper functioning, and this is the xylem. So the xylem is taking the water and minerals. Now these mineral ions will come out of the xylem, right? Mineral ions will come out of the xylem by diffusion. These are the lines of NCERT. At the root, there was a mixture of water and minerals, right, in the xylem of the root. From the xylem of the root, this mixture is going up with the help of transpirational pull in the xylem of stem. Then this is now present in the xylem of the leaf. This is the xylem of the leaf containing minerals. Now the sink cells, that is the cells of the leaf, they need the minerals. So these minerals will come out of the xylem by diffusion, right? This is the line of NCERT. These sink cells will take these mineral ions actively, because beta, these sink cells, they need mineral ions and they are living cells. So when they will absorb the mineral ions, that would be the active process. So remember, when the mineral ions will come out of the xylem, because xylem is a dead tube, and dead things, they do not restrict the entry and exit. So xylem is a dead tube. The mineral ions will go out of the dead tube by diffusion. But when these sink cells, the living mesophyll cells, when they will take the mineral ions, that would be active process. Okay, so unloading of mineral elements from xylem. Unloading means when the mineral ions will come out from the xylem at fine endings, it is done by diffusion. This is the line of NCERT. This is the as it is line of NCERT, that unloading of mineral elements from xylem at the fine vein endings. Vein endings, wherever there is vein, there is xylem. So unloading of mineral ions from xylem at the vein endings occurs through diffusion, but the uptake of mineral elements by sink cells is active. Okay. Now there is another point in NCERT, remobilization of mineral elements. What is remobilization? Remember that some mineral elements, they move from old leaf to the young leaves. That occurs through phloem, and that is called as remobilization of mineral elements. This is the old leaf, the old senescent leaf which is about to die. This will donate its element to the young leaf, and these minerals from old leaf to the young leaf will go not with the help of xylem but with the help of phloem. Okay, and the elements which can move from old to young leaf, they are called as the mobile elements, but those which cannot move and they will go along with the old leaf, they are called as the immobile elements. So this is important, that elements which can be remobilized from old to young leaf, they are mobile elements like nitrogen, potassium and phosphorus. But those which cannot be remobilized are immobile elements like calcium and sulfur. And remember, this is important, that in one chapter in NCERT it is given that sulfur is mobile, and in another chapter in mineral nutrition it is given that sulfur is immobile. In this chapter they are saying that sulfur is readily mobile. False. False. Sulfur is immobile. You have to remember this. In this chapter, it is given that sulfur is readily mobile. This statement is false. Calcium and sulfur both are immobile. Okay, in NCERT it is given, this is false. Okay, beta. Now, there is a notion, usually what people think, that minerals are only translocated through xylem. Wrong, I have just proved it. Minerals are also translocated through phloem also, when they are moving from old to young leaf. First thing. Second things, it is thought that minerals are translocated through xylem only in inorganic form. This is false. This is thought that inorganic molecules and water move through xylem. No. Through xylem, the mineral elements, they move both in organic and in inorganic form. This is very important. Lines of NCERT. That minerals translocated through xylem both in organic form and in inorganic form. Clear? Like nitrogen. Most of the nitrogen through xylem is transported in organic form, in the form of amino acids and amides. This question comes directly, that most of the nitrogen in xylem is translocated in inorganic form or in organic form. So it will be, answer will be, mostly it is translocated in organic form through xylem vessels, and only some of the nitrogen is translocated in inorganic form. But this is opposite in case of phosphorus and sulfur. Most of the phosphorus and sulfur are translocated in inorganic form, and some of it is translocated in organic form. But the thing is that the mineral elements are translocated both in organic and in inorganic form through xylem. Clear? So this is important. Okay. Now let's discuss transport through phloem. This is important. Up till now we have discussed translocation through xylem, but now let's discuss translocation through phloem. Okay, beta. I hope things are clear up till now. Let's discuss the phloem transport now. We know, beta, that the transport in phloem is bidirectional or multidirectional, right? Because the source to sink relationship in phloem is not fixed. I've just told you it changes seasonally. What happens that in when the autumn season comes, the leaf fall, and when there is leaf fall, the root become the storage organ in winters. Then spring come. During spring, the root provides the food to the different parts of the plant and helps new leaf to come and flower to come. And when the spring will end, the plant will be filled up with leaves, and again the leaf will become the source. So the source to sink relationship is not fixed. That is why it is bidirectional or multidirectional in case of phloem. Clear? So what actually gets translocated through phloem? The mixture, the solution which is present in the phloem is called as phloem sap. Now, what is the constitution of phloem sap? That makes an important question. The main constitution of phloem sap is water and sucrose. What is mainly present in phloem sap is water and sucrose. But along with it, other soluble sugars, hormones and amino acids are also present in phloem sap. This is important. If the question says main ingredients of phloem sap, your answer will be water and sucrose. Then if it asks the overall composition of phloem sap, this. Clear? Okay. Now this was the question of NEET, that the nature of phloem sap is slightly alkaline in nature. So these things need to be remembered. Now let's see what happens at the source point. This diagram is very important and listen everything carefully. Now let's understand. This is, understand very carefully line by line. This is the mesophyll cell. Okay, this is the mesophyll cell. Mesophyll cell is the cell in which food is formed of leaf. This is the mesophyll cell of the leaf. In the leaf, photosynthesis has occurred, which resulted in the formation of glucose. So in the mesophyll cell, because of photosynthesis, glucose is formed. Now remember, the food is not translocated in form of glucose, because glucose is a reducing sugar, reactive sugar. So first it has to be changed into sucrose, because sucrose is a non-reducing sugar. So the first step is glucose is converted into sucrose inside the mesophyll cell. Then this sucrose comes out of the mesophyll cell. This is active. This takes energy. Step by step. Mesophyll cell, photosynthesis occurred, glucose formed, glucose is changed into sucrose, sucrose is taken out of the mesophyll cell, and see what is written over here. Unloading of sucrose from mesophyll cell is active. To take sucrose from mesophyll cell out, energy is needed. This is active process. Then you know what is a phloem, right? A phloem is composed of companion cell and the sieve tube. So this is the companion cell and this is the sieve tube. These are the sieve plates and these are the cytoplasmic strands present in the sieve tube. And unlike xylem, sieve tube is not a dead tube. Sieve tube is not a dead tube. You have plasma membrane, cytoplasm and cytoplasmic strands in it, right? So this is the sieve tube. Clear? Now this sucrose will load into the companion cell. The sucrose will load into the companion cell, and the loading of sucrose in companion cell is again active. When this sucrose will come into the companion cell, this will be active, needs energy. Then from here, this sucrose will be loaded into the sieve tube. And again, this is active. Loading of sucrose into the sieve tube is active. Are you clear with this now? Up till here, it is clear. So what has happened? Generally, the source is leaf. So here we are taking leaf as source, and inside leaf glucose has formed, right? Changed into sucrose, which is a non-reducing sugar. This sucrose came out of the mesophyll cell, active process, loaded into the companion cell, active process, then loaded into the sieve tube, again active process. Clear? Now what happens, beta? Now what happens? This sucrose is loaded into the sieve tube, right? Now imagine what is happening inside the sieve tube. This is sieve tube. What is happening inside the sieve tube? Because of accumulation of sucrose in the sieve tube, what happens? Hypertonic solution gets formed inside the sieve tube. What I have just told you, sucrose is accumulated inside the sieve tube and sucrose is a solute. Because of this, because of the accumulation of sucrose into the sieve tube, hypertonic solution is formed in the sieve tube, because of which the water potential of sieve tube decreases. And you know that wherever there is phloem, there is xylem besides. In a leaf, xylem is present besides phloem. So this is phloem, in which the water potential is decreased. So this sieve tube will go to xylem. This sieve tube will go to xylem and say, please I want some water. Lend me some water. I want to borrow some water from you. So this xylem, this is the xylem. This will give water to the sieve tube because xylem has enough amount of water because it is coming from the root to it. So xylem has enough amount of water, but in the leaf, the sieve tube, because of accumulation of sugar in it, its water potential got decreased. So it has borrowed water from the xylem. The water from the xylem moves into the sieve tube. And because of this, because of the movement of water from xylem into the sieve tube, the turgor pressure at the sieve tube becomes high, right? And imagine the sieve tube of the leaf where the water has just filled up into the leaf and the turgor pressure has become high. But what is the condition in the phloem of root? In the phloem of root, in the sieve tube of the root, the water is not there. So the pressure is low. So what you have just created? Pressure gradient. In the sieve tube of leaf, there is high turgor pressure, right? But in the sieve tube of root, there is low turgor pressure, and you have created a pressure gradient between 2 points, and this is mass flow. This is mass flow. So in the sieve tube, high turgor pressure is created, and because of this high turgor pressure, the things will move by themselves towards the root. Clear? Are you clear with this? Now, what is happening? You have created, what is actually happening? You have created high turgor pressure at the source, and there is low turgor pressure at the sink. And right now what we are taking as the source? We are taking leaf as the source and root as the sink. That is the different case, that it can change, but I am exemplifying with the help of leaf and root, right? So at the source point, the turgor pressure is high in the sieve tube, but at the sink point in the root, the turgor pressure is low in the sieve tube, and you have created what? You have created pressure gradient. Due to this, mass flow of food occurs, and this is called as mass flow or pressure flow hypothesis. And this movement of phloem sap in the phloem is also helped with these cytoplasmic strands, because when the phloem sap will be moving across the sieve tube, it will not be epoplastic movement, it will be symplastic movement, because the sieve tube is not dead. The water across xylem is epoplastic because xylem is dead within, but the movement of phloem sap inside the sieve tube is symplastic. That doesn't make it active. It is done with the help of mass flow, but it may be helped by the cytoplasmic strands also. Clear? Okay. Now let's come to the sink. Now the phloem sap is flowing. Now the phloem sap is flowing it has come down to the sieve tube of the root. Now the phloem sap is present in the sieve tube of the root. This is root setup. This is the setup of root. This is the sieve tube of the root. Now sucrose will come out of the root. Sucrose will come out of the sieve tube, and this is active. When the sucrose will come out of the sieve tube, this is active. Energy will be needed. Then sucrose will come out of companion cell. This is also active. Then this sucrose will be taken up by the sink cell, that is the root cell. This is also active. And what will happen when this sucrose will unload from the sieve tube at the sink point? Now what is happening at sink? Inside the sieve tube has given its sucrose out to the root cell. because of which, because of the unloading of sucrose, from the sieve tube, hypotonic solution is formed. Hypotonic means sucrose, sugar has gone out. So the concentration of the solution got reduced and it has become hypotonic solution, and the water potential inside the sieve tube increased. Why? Because sucrose went out. And because of this, the water potential in the sieve tube increased. And remember, at the leaf, the sieve tube borrowed water from xylem. Now this is the time to give back that water to the xylem. So at the sink point, yani ke at the root, this sieve tube, because the water potential in the sieve tube has become high because the sucrose has gone out. So the sieve tube will return the water it has taken from the xylem. And it is done by osmosis. Are you clear with this? This actually happens during the translocation of food. Let me recapitulate it again for you. Let's go to leaf. Imagine with me. Let's go to leaf. In the leaf, mesophyll cell is present, in which glucose is formed by photosynthesis. It is changed into sucrose. This sucrose will come out of the mesophyll cell, active. It will load into the companion cell, active. Energy will be consumed. Then it will be loaded into the sieve tube, active. Because of accumulation of sucrose into the sieve tube, the hypotonic solution will be formed inside the sieve tube. This will decrease the water potential of the sieve tube, and the sieve tube will borrow water from the xylem. Because of this, the turgor pressure becomes high at the source, and at the sink root, turgor pressure is low. So pressure gradient, mass flow will happen. Then what will happen at the sink, at the root cell? The sucrose will come out of the sieve tube, active. Then it will come out from the companion cell, active. Then the sucrose will go into the sink cell, root cell, active. Because of the unloading of sucrose from the sieve tube, the water potential of sieve tube increases, and the water goes out of the sieve tube into the xylem. This is how translocation of food occurs, right? When water goes from xylem to phloem or phloem to xylem, it occurs by osmosis. And remember, at the point of source, turgor pressure will be high, and at the point of sink, turgor pressure will be low. Clear? Then finally comes the Girdling experiment. What is the Girdling experiment? This is the experiment which actually proves that the translocation of food does not occur through xylem but through phloem. And what we do in this experiment, we take a plant, right? We take a plant and we remove a thin ring of bark. What is bark? The detail of this we will deal in anatomy. Every tissue which is present outside the vascular cambium is bark. That means phloem also comes in bark. What does not come in bark is xylem. The central portion does not come in bark. Whatever there is outside the vascular cambium, that is bark. That means if you remove bark of a tree or a plant, you will remove its phloem. So I have taken a plant and I have removed bark from this portion, only from this portion. Okay. So after some point of time, what will happen? Some material will be accumulated over here. And when I checked this material, what I found out that sucrose is present in this material. That means I have not removed xylem from this part. If food have been translocated through xylem, then sucrose would not have been accumulated over here, because we have not removed xylem. The continuity of movement of food would have been there, but it is not translocated through xylem, because of which the sucrose got accumulated here. because we have removed phloem from it. So this proved that the translocation of food does not occur through xylem but through phloem. So girdling experiment is the proof of it. And I think in NCERT it is written that girdling experiment proves that the translocation of food occurs only through phloem in one direction. That one direction is false, right? Clear, beta? So with this we are done with the chapter, and I hope you all understood the chapter very well. Just go through the lines of NCERT, beta. After having listened to the lecture, go to your NCERT, read your NCERT lines line by line. I have dealt each and every line of NCERT with, and then after doing that, do as much questions as you can, right? What we have discussed: facilitated diffusion, plasmolysis, uptake of water is passive, uptake of mineral ions is both passive and active, but mostly active, how it is done, right? How translocation of food through phloem occurs, what is mass flow, transpirational pull, root pressure. Okay, just if you don't get things once in one time, just pause the lecture, rewind it back and listen it properly. You will get the thing, right? I had to be a bit fast in the lecture because it would not have been completed in one shot, but I hope you will understand the chapter. Thank you.